DNA sequencing is a process by which you can determine the order of nucleotides in a DNA segment. The most common form of DNA sequencing is called Dideoxy sequencing, and it is done in 4 separate tubes and 4 separate reactions. Each tube contains a same copy of the DNA segment, free normal deoxynucleotides, and a primer and DNA polymerase. Okay, and so also in each of these tubes is something called a dideoxy nucleotide, and each one has there are 4 different ones, and each one has a specific one. They're called DDA, DDT, DDG, DDG. They are different from normal nucleotides in the fact that they, instead of having they are missing a 3' hydroxyl end of the nucleotide, which makes them unable to form bonds with other nucleotides. So it stops the annealing of nucleotides when the primer inacts. Okay, so in DNA sequencing, in each tube, the primer will attach to the end of the complementary strand of the DNA, and the DNA polymerase will begin adding nucleotides to each strand. As the strand goes on, when it reaches, like, say you have DDA in one of the tubes, once it reaches a T in the complementary strand, the DNA polymerase has an option of adding either a normal A nucleotide, or it has the option of adding a DDA, which is and because there's only less than 1% of the concentration is DDA, it won't happen most of the time, but it happens enough that all the T's get coded for the DDA in each section, because it happens millions of times over. But anyway, so if it does add DDA, I believe we were talking about T. When it adds a DDA, the coding is stopped there, and the next one moves on. It's the same for all DDT, DDG, DDG, DGC. It does that for all of them. Okay, so once it does that, you run a gel electrophoresis through the samples, which determine the length of the strand. The shorter ones go to the end, and nowadays they use something fluorescent tags on the special dideoxy nucleotides, a fluorescent tag, so that you can read it. And when you read a gel electrophoresis, you read it from the 5' end to the 3' end. So that would be, depending on if you use your complementary strand or your normal strand. If you use complementary, what you're reading is the actual DNA strand. So, there.